All right, hi class. How was life? That good, huh? How many people have more than 2 tests this week? More than 3? That counts. Okay, so you're feeling like you're getting the true experience. Excellent. Also, do people know we only have like 5 weeks of in-person class left? Which is not very many. We're like two-thirds done. Yeah. Anyways, this October is a rough time because, I mean, it's a wonderful, magical time, but it's rough because I think people sometimes feel like they've been in school for a long time and there's still a long ways to go, and that's hard. But there's not that much longer to go. I mean, there is, but not that much longer to go. Like, by this time next month, you'll be thinking about, I don't know, knitting decorative socks for your loved ones for Christmas or whatever it is you do, right? Anyways, okay, things to remind you, which, again, the group that's here is not the group I'm most worried about. You have an exam on Friday. You do not need to come to class in person. It'll open early, it'll close late. Don't cheat amongst yourselves. You will fail the assignment if I catch you doing that. I wouldn't have said this except for we had a faculty meeting yesterday, and we had a whole bunch of things to tell people about stuff. What's that? This week for lab, you're doing your self-designed lab experiment. Most people that I've talked to are doing it in their dorms or outside or, I don't know, trying to hypnotize the geese or whatever it is you're doing outside of lab. If you need to be in lab, nobody's actually doing it, but I want to see it one of these days. It'd be so cool. If you need to be in lab, that's cool. If you need stuff, let me or Robin know before, like, the moment of. You don't turn anything in for that one, except this project at the end of the semester, you'll have your brief, like, 5-minute PowerPoint of what you did. So remember to take pictures if it's something cool that would help. You don't need to take pictures if you don't want to. And you'll be using that to feed into your paper that you're writing for lab. That first draft of the paper is due next Wednesday. Okay? Robin sent an email out about this. Yes? People got this? It's kind of a bare bones draft of your paper. You might not have everything done or ready yet. That's fine. But you at least need to be working towards it. List of things to remind you, tell you about. Oh, other thing, after class, as soon as I get around to it, I will put up another, if you want to write a couple questions to maybe have appear on the exam, you can do that for extra credit, if you like. That will go up today, and it'll close by Thursday, like 5, because, you know, you have your exam on Friday. So far, so good? Sweet. Yep, we're good. We were here, right? Okay. So we have our cell, it's just doing life right here. It's synthesizing DNA here. It's in G2 right here, which is the step between synthesis and actually getting going. This is often fairly short. And then going to mitotic phase where it's gonna organize the duplicated or the replicated DNA, organize it, tear it apart, break that into 2, and you're good to go. Questions are going to have a payoff later. If you were trying to control this system, if you're trying to have this system and be like, you know what, I don't want it just to wildly be cranking out cells. I mean, question, do you want this to be wildly cranking out cells? Why not? Because, like, more cells are better, right? Yeah. Yeah, I mean, yes, amongst other things, yeah, absolutely. If cells are wildly duplicating for no apparent purpose and they're not supposed to be, that's the definition of cancer. Just for the record, I'm going to reference cancer several times today. I legitimately recognize that probably everybody in this room knows somebody either with cancer or who's had cancer, and it's somewhat a fraught topic, right? And then there's also always the like, well, if science knows what's going on, why hasn't science fixed it, right? The problem is, cancer as a whole, yes, you have abnormal cell growth, but what causes that, what's controlling that, there's a whole, cancer isn't one disease, it's a description for a whole bunch of different things, all of which are having the cell cycle be messed up. Okay? We're okay with this? Okay, so, great. So we've decided we don't want this to just turn out all the time. You want this to happen because you want cells to be able to divide, but you don't want this going nutso, right? You also don't want it to go nutso just because it takes a lot of energy to, like, you know, support cells. And you want the cells you need, you don't want extra cells. I mean, you want some extra cells. You don't want wild growth that you don't need. If you were trying to control the system, because, the power, control, whatever, where do you want to put, you know what? Stop and check in with me before you move on to the next stage. Where would you put that in here? There's like, I'm legitimately just curious. Where would you put that in this system? After synthesis? So you're going to replicate DNA and be like, okay, do we really want to go through this? Is that fair? The cell does that, actually. Any other spots people want to have a control point? Before we replicate DNA, as Noel says, like, okay, we're going to replicate DNA. We want to make sure, are we sure we want to do this? But Jesse's point is, even before we replicate DNA, like, whoa, it's going to take a lot of energy for me to replicate DNA. I want to check before I get there. There's one like that, too. So that one's the most important one. There's another one, here-ish, and then there's one here as well. We'll get to it. There are 3 control points, and they're going to do different things. But yeah, this cycle is under, in the absence of cancer, it's under fairly tight control, okay? Just because I feel like I need to show you the same pictures over and over, and I got excited and looked up online for a whole bunch of little time-lapse videos, and I like time-lapse videos, you had the same pictures many, many times. This is where we were, though, just to remind us what was happening. I know you saw this, where you have, by the time you've gotten to G2, you've already duplicated your DNA. A really, really common misconception is that this mitotic phase, this M phase, duplicates DNA and then splits DNA. No. It does the splitting part, but the duplication has already happened, okay? That's happened in the synthesis phase. So you have this duplicated, kind of tangled up chromosomes. You're going to start getting them organized. You're going to pull them apart. You're going to line them up. You really need to make sure that you get an even amount on both sides, right? You know how, maybe not. Anybody here do the thing at Halloween where you and your siblings or buddies dump all your candy in the middle, and then you have an elaborate trading system where you separate it out? Okay, like there are rules, right? Okay. Does anybody want to describe that to the class? I feel like 3 of your faces were like, yes. Haley, do you want to try? If this is pressure, you don't need to.
I mean, like, our system is if someone has one candy that's theirs, like that is their favorite candy, they get all of it.
Oh wow, nice.
Oh yeah, there's no issues with it.
Nice. What?
Even if this sibling likes it,
too bad.
That one loves it, so that one gets it.
You have one. Okay, perfect.
We have one sibling that we just kind of give all the bad candy to. Like they don't get a choice in that.
Are they okay with this?
We don't care.
Who knows?
There are 6 of us. We don't care.
It's like, listen, sorry. Are your rules slightly different?
Yeah, 'cause it was just my sister and
Uh-huh.
I, and we just trade stuff.
Trade stuff. Okay, fair enough, fair enough. Okay. If you wanted to go for the communist method of this, which I realize nobody does, right? What you do is you dump all the candy in and you'd like one Smartie on this side, one Smartie on this side. One Twix bar on this side, one Twix bar on this
That's
side.
what my 2 little siblings try doing.
Right, okay, if you're like, it will be exactly fair, right? Cool.
Then the rest of us fix it.
Okay, gotcha. So the communist method, right, is where you're just gonna split it down the middle, okay? Cells really want to do that, okay? Cells want to make sure that if you get a chromosome 1, I get a chromosome 1, right? You need this to happen. It's worth noting that a cell, before this whole thing goes down, let's say you have a cell that has 3 pairs of chromosomes, okay? So your cell has 3 pairs of chromosomes, meaning it has a chromosome 1, a chromosome 2, a chromosome 3 from mom, and it also has a chromosome 1, a chromosome 2, a chromosome 3 from dad, okay? Chromosome 1s should have the same genes on them. So chromosome 1 has the genes for hair color. The one from mom has a gene for hair color. The one from dad also has a gene for hair color. They might be slightly different versions, but they both are coding for the same thing, okay? You've got one roll of Smarties, you've got another roll of Smarties. This roll of Smarties might have 1 or 2 more of those like light green Smarties, and this one might have mostly white Smarties, but they're both tubes of Smarties. Rolls, tubes, I don't know, right? Are we okay with this? They're the same, they're called homologous pairs, but they're the same type of chromosome, okay? So if your cell has 3 pairs of chromosomes, it's got 6 chromosomes. When it's at this phase, like here, metaphase, or anywhere after it's replicated, your 3 pairs of chromosomes, each of those are a pair of sister chromatids. So if we count actual like little stick parts, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? We're gonna pull that apart, so each cell is gonna get 6, which is 3 chromosomes from mom, 3 chromosomes from dad. Are we okay with this? Great. So then we pull it apart, we reform the nucleus, we pinch that in half, and it's beautiful. And how come I can't click buttons? Really fast, just so you see it. It's kind of cute. That's the chromosomes there, and you can see the spindle fibers attaching. So there we go, things are attached, they pull apart, they pinch off. If you want to watch that really fast, this one's way too fast, and I don't know how to embed it and slow it down, and I should look this up, but I actually don't know how. Yet. I might learn before the end of this year's up. It's just kind of cute. Pachoo! Right? Yeah. And then okay. What's worth watching here, what we're going to see is cell division. But what's happening is a cell's getting fertilized. So you're going to see you've got like one cellish. You've got you've got 2 gametes coming together. So you've got like an egg and a sperm. They're going to come together. You're going to see the 2 separate nuclei meet up, right? So fertilization is going to happen, and then the cell needs to make more of it, right? The plan is for growth. So you have your 2 separate nuclei. They're going to come in together. You're going to dissolve the nuclear membrane, right? You're going to organize the hub, dissolve the nuclear membrane. You don't actually get to see chromosomes here, but you're going to see the nuclear membrane dissolving, and then you have 2, right? So it's going to dissolve, pull apart, reform. It's going to do that again. It's going to dissolve, reform, pull apart, and then you end up with 4. So you see that here, and you can watch it. actually make this bigger. Then they fuse. The picture on the left is looking at the nuclear envelope. You see two of them come back. You need to dissolve that up again. This one's actually faster at it. It's forming two more up here. This one's trying to get going. Do we see how that's happening? Okay with this? And it's going to keep going. I mean, during development, this just happens quickly, right? You go from 1 to 2, 2 to 4, 4 to 8, 8 to 16, 16 to 32, 32 to 64, right? You get bigger faster, right? Like, boom. Okay. Now we're through here, through here, through here, here, through here. This is mitosis in an animal cell. You can go ahead and watch it, because I think they're pretty. So can we all see the cell right here? We're looking, here's we're looking. This is basically the nucleus. And then we've got actually a fair bit of the cell out here. I realize it's kind of hard to see. You can see chromosomes in the middle, though. And you'll see them condensed down and try to get themselves organized. And you'll also see the nuclear envelope go away. So, like, you can't see the nuclear envelope anymore now. You see how the chromosomes kind of got themselves organized a little bit? They're trying hard. Because you see how now we've pulled everything to the middle, even though some of them weren't originally at the middle? Boom! Once you get everybody to the middle, you tear them apart. We're okay with that as a picture? And I have two separate distinct cells, right? There's one cell edge here, another cell edge here. We've got a nucleus that's been formed up here, been formed up here. You see that? We're okay with this? And actually bears noting, like, cells are supposed to do stuff. Their mission in life isn't to just replicate. They're supposed to be doing something. So, like, yeah, during development, usually they're just replicating really quickly. But typically, these cells are doing some cell job. And they're replicating to make more of them. And you've already seen this. This is just, again, what we saw. You're expecting these chromosomes, these sister chromatids, to get lined up in the middle, and then the whole thing to get pulled apart. And I really got excited about this because I found a website that had a bunch of these, and so I got very excited. This is just looking at the spindle formation during mitosis. It's really kind of fast, so we'll look at it. See the spindles forming, and then they're going to start pulling apart. Yay. Some terms, I'm going to skip that, but here we go. So cytokinesis, I know we touched on it on Monday, but I wanted to yap about it more. Just briefly. So if you're looking at this animal cell, you'll see how you've got plasma membrane around the whole thing, right? You end up with a little I completely blanked on the word. Protein. Anyways, there's a specific name for it. Microfilaments. Microfilaments, what it is. You have microfilaments that come and like squeeze it off. And so you end up with these two cells, each half the size of the other one, and there's plasma membrane around the whole thing. Okay? In plant cells, it's a little bit different. You'll see here on, let me actually make it so. Just a sec. Sure, I think that's actually viewable. Okay, but I have to talk loud now because of this. You'll see how, yeah, see how right out here we've got a plasma membrane along this whole edge of the cell? Cool. These are vesicles, and they've got plasma membrane on the outside of them, and inside this vesicle, it has goo to make the cell wall of a plant. So you line a bunch of these vesicles that have plasma membrane on the outside, right? They're vesicles, they're bags of plasma membrane. Inside they have the cell wall goo. You line a bunch of them up, smoosh them together, and by the time you have this smooshed together, the cell plate that formed up forms, so again, you've got plasma membrane on the outside, goo for cell wall in the middle. Now we've got plasma membrane all around the outside, goo for cell wall in the middle. You run everything together, and you've now just made a new cell wall down the middle. We see how that works? Okay with that? Here is the exciting video for it. Online people, watch this in your notes because well, let's see. The other ones haven't had sound. I want to see if I can make them play at exactly the same time or it'll echo horribly. Oh, come on. I'm not coordinated enough to really pull this off well. Okay, fine. Online people, no.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Okay, so far so good. And here's another video just because they're so cool to look at. This is just looking at cytokinesis happening. Yay! Right? It's cool. Just have a couple more pictures to show you. I was mentioning before class, the backstory for how science found this was just somebody named Dr. Fleming, late 1800s, was looking at cells and noticed, when I stain for what we now know are chromosomes, when I stain for chromosomes, I first see it all over in the mess in the cell, well, in the nucleus, it's all over, and I see it, this pattern always happening. So whether I'm looking in onions, whether I'm looking in animal cells, I'll see it where they'll be all over, they'll be condensed down, they'll line up in the middle, they'll pull apart. And I keep seeing this pattern over and over. When you see patterns over and over, you're like, maybe I should pay attention. So here we are. This is plant cell. How can you tell it's plant cell? The wall, yeah. We've got prophase here. We've got prometaphase. You can see them starting to condense down. Metaphase, you see them lined up. Anaphase, they're starting to pull apart. Telophase, they're starting to, you're forming up the cell plate, and you're done. Okay, so we've done eukaryotic cells for like half of today, right? And all of Monday. We're now going to look briefly at prokaryotic cells. So we've done eukaryotic cells, we're now at prokaryotic cells. So prokaryotic cells, which is basically bacteria, but also archaea, they're going to reproduce by binary fission. So this is cellular division. And again, it's cellular division that's this mitosis version. It's you're not getting some genetic material from mom and some genetic material from dad. No, You have your cell, boom. Wherever you start with the cell, you're replicating that DNA and pulling it apart. Just like mitosis is working for you, right? You have a cell in your arm. It's gonna replicate its DNA, pull apart. Now you have two cells. Okay? Bacteria are different when they do this than eukaryotic cells because they don't have a nucleus, right? They don't need to dissolve this nucleus to pull this apart. It's really fast. It's really, really fast for bacteria to divide. For multicellular organisms dividing one cell doesn't make a new organism, right? It just makes a new cell. If you're a single cell organism, if you divide, you suddenly have two organisms. So it looks like this. Bacterial DNA, you're expecting it to be in a ring, like a tire inner tube almost. DNA is nice and double-stranded. You open it up and you start to replicate that DNA. You end up making two copies at each end of the cell. Get two ring circle situations of DNA. And then you don't have a nucleus to have had to dissolve or to reform, so you just go ahead and you pinch that off, and now you have two cells. Just like eukaryotic mitosis, the DNA that you're expecting up here, right, from the parent cell, you're expecting the DNA down in the daughter cells to say the same thing. You might make mistakes sometimes, but you're expecting it to be a copy. All right? Just so we're all clear on terms, this original cell is called the parent cell, and then the two cells that are made are called daughter cells. There aren't son cells, I don't know why, but it's the parent cell and the daughter cells. But you can have parent and daughter cells, and it's not actually talking about your physical parent. It's saying the original cell we're talking about, duplicating its DNA, pulling apart, now daughter cells. Everyone's okay with this? Questions? Okay. It gets tricky because once we start about sexual reproduction, it's wait, when you say parent cell, what do you mean? Okay, so this is pretty straightforward, right? And the eukaryotic version is pretty straightforward too. There are a couple, can you go ahead and pull it up, thanks. There are a couple weird versions too. Okay. So, bacteria have the basic, what looks like the original version of doing this. You replicate DNA, you pinch your cell in half, right? Eukaryotic organisms have a fairly straightforward version of okay, we're going to dissolve the nucleus first, pull those chromosomes apart, and then pinch the cell off. Cool. Some organisms, they're called diatoms, and some yeasts have a version of this that's a little bit different. They're okay, fine, but I don't want to dissolve my nucleus. That seems like a lot of work. So I'm going to keep my nuclear envelope intact, but make it big and spread it apart. And so I spread this whole thing, and when I pinch this off, I'll pinch off the cell membrane and the nuclear envelopes at the same time. Right? We're okay with that? Makes sense. Not super common in biology, but it can do it. And then there are dinoflagellates, who apparently did not even read the manual, and that's fine. But they apparently, and again, so this is not actually, but it looks like they kind of were listening to the other cells and being okay. I need to separate chromosomes. Right. Separating chromosomes. I need to make microtubules. Okay. And I need to pull this part to each side of the cell. Okay, I got that. And they weren't really focusing. And so they do this weird, freaky thing where they keep their nuclear envelope intact, but they run microtubules through it, and this thing is full of holes, and then it pulls it apart, and then closes up, and then you have to seal the holes back up and get rid of microtubules. It's a mess, and it works for them. Okay? You've seen people do this though, right? Like, whatever, like they're trying to save a PDF file or something, and so they like they email it to themselves, then do some like weird convoluted thing where then they open it up on a website, and then they download it and they send it themselves via Dropbox, and you're like, I don't know what happened, but if it works for you, let's just go with it, right? The basic versions are either binary fission or normal mitosis, which is what eukaryotic cells do. We're okay with this? I do like dinoflagellates. They're these little protists. They're kind of adorable, and they're really weird. Okay, last 20 minutes is actually the conclusion of some of those questions at the beginning of how do you control this thing. You control it molecularly. Molecularly is not a word, is it? Anyways, too many syllables. You control it via chemical signals, all right? And you want different cells to divide at different speeds. Any cells in your body you think should divide pretty quickly? Skin cells, right? You're forever doing something to your skin, right? You're forever, I don't know, scratching your arm or picking something up or whatever it is that you're forever damaging these cells, right? Skin cells divide really, really rapidly. Stomach cells, like stomach lining cells, oh my goodness, you're eating all sorts of random junk. Things are always moving, things are scraping up against that side. The system is full of acid, those cells just have a hard time, so you keep dividing those. Some cells, say neurons, you have one. Once you have it, it's not planning on dividing, typically. It's planning on just being there, right? They're going to divide at different times. You also, at different times through an organism's life, at different times it's expected to behave differently, right? So if you have a single kernel of corn, right, you're expecting that cell and then those cells to behave differently at different times, right? So if it's just sitting in your nice little seed packet in your freezer where you're keeping it, you're not expecting it to divide at all, right? You're expecting it for it to wait until you have some environmental cues, right? You want it to be warm, you want it to be wet, you want it to maybe have some dirt around it, and then you want that cell to respond. Fair enough? Cancer is going to be cells that have escaped the normal controls on the cell side. There are a couple controls. So it looks like this whole cell cycle, it looks like it's controlled by specific chemical signals. Those chemical signals can come from within the cell, or they can come from outside the cell and come in. You can actually, a clever experiment, it's a classic now, is this. So, we're going to go back up just a sec. Here. We're all clear, you're expecting G1, S, G2, then mitosis and cytokinesis. We're solid on that, that's what we're expecting. Okay, so now, back to where we were. Here we go. Okay. So experiment 1, you have a cell that's in S phase, you have a cell that's in G1 phase, and you squish them together, right? When you do that, the cell that's in G1 phase immediately goes into S phase, immediately starts synthesizing its DNA. The thought is, okay, probably there's some signal here in the cytoplasm of this cell that's in S phase that's saying, be in S phase. Go ahead and synthesize your DNA. So when you smush them together, this cell had been chilling at G1, now knows I should synthesize DNA, right? That's suggested, but you don't actually know that. 'Cause maybe it's just the time for the cell to go into S phase, because after G1, S phase happens, right? So the more compelling version of this experiment is you have a cell that's at M phase. Remind me what happens at M phase? So yeah, so you've already duplicated it, right? Oh, sorry, you've already duplicated the DNA, but you're making 2 cells now, right? Yeah, perfect. Okay, we're okay with this? We're gonna split that apart? Okay. This cell's at M phase. You're expecting to pull this apart and then, cut it in half, right? We're okay with that? If you smush the cell that's in M phase with the cell that's in G1 phase, you smush them together, the nucleus of the cell that had been in G1 phase goes into M phase. It starts building microtubules and the nuclear envelope starts dissolving and it is ready to tear itself apart, right? This is a problem for this particular cell because it has not synthesized another set of DNA, right? This one over here, this first one that had been in M phase, was ready to go, right? It already replicated its DNA. This one over here in G1 hadn't had S phase happen, right? It's not happened yet. So here, when it goes into M phase, we can see that, you know what? It's not the series of events happening that makes the next stage happen. It's a chemical signal that tells the cell what to do, right? So even though there aren't enough chromosomes, the chemical signal that was over here in this cell saying, hey, pull yourself apart, touches this one and says, hey, pull yourself apart. And so it's ready to do that. Do we see that? The take home from that is there are chemical signals that tell the cell what to do, right? Which is really good news because the cell cycle is controllable. It's bad news if it breaks. But this whole system ends up with what are called checkpoints. And it looks like this. So there are 3 checkpoints and 2 of the major ones. I mean, there are 3 major checkpoints. And they're at where you all kind of guessed they were going to be. So you've got one called the G1 checkpoint. It's in G1. You have one that's a G2 checkpoint. We're not really going to worry about it. And you have another one called the M checkpoint. So the G1 checkpoint tends to be the most important. If cells get the go signal here, they're going to synthesize DNA, and typically it's going to complete the S phase, the G2 phase, the M phase. Typically, if they go here, they go all the way. If they don't get the go signal here, they can exit the cell cycle and just chill in a non-dividing state called the G0 or G0 phase. Okay, so the cell's in its cell cycle, right? It doesn't get the signal to go here at G1. It's like, you know what? We don't need you to divide. Just be you. Just be a cell. Do your thing. Okay? This is great. It's not going to spend extra energy trying to synthesize more DNA. It's just going to be the best little cell it can be. Okay? If there's a signal that says go, the cell cycle keeps going. And at this point, you're expecting this to be carried to completion. So the cell goes. It gets to, there's a G2 checkpoint. We are going to ignore it because minor details. The one that's also important is the M checkpoint. This one, you've gone to the point where you've had DNA synthesized, you've had it hopefully condensed. The M checkpoint is waiting for the signal that every single chromosome is attached. All right? So if you're going for the dumb analogy thing, I don't know, what someplace as a child you wanted to go, enjoyed going and visiting. A library, amusement park, I don't know, ice cream shop. Someplace you're like, yay, park. Let's go for a park, okay? You can go to the park. Your mom or dad or grandma or whomever is like, okay, if you get the go signal to get your shoes, get in the car, you're gonna go, right? Okay, that would be the G1 checkpoint. They're like, by the time we have everybody's shoes on and we're heading to the car, we're gonna go all the way, we're committed, right? This M checkpoint is a lot like responsible adult figure not taking their foot off the brake or actually not even turning on the car until, are you all buckled back there, right? Like, we're definitely gonna go, but I'm not going until everybody is safe. Fair enough? The cell has this M checkpoint that's, we're gonna do this. I'm not doing this until I have all the chromosomes attached. Because otherwise, as you tear this cell apart, bad things happen. Fair enough? So the G1 checkpoint's really important for, are we going to divide? The M checkpoint's really important for, are we going to divide appropriately? Fair enough? Great, so I will ignore this one. Here's where we are. Cell division influenced by lots of things. So growth factors are going to encourage it. So you might have heard of something like called human growth hormone. It's a hormone that promotes human growth. Yeah. It's going to be released by certain cells, and other cells receive that signal and are like, oh, you know what? I should move past my G1 checkpoint. I should really divide. Gosh, I'd like to do that, right? You can have growth factors caused by platelets. So like, if you have tissue damage and you end up with blood cell fragments, right? Something has gone down. You've got chunks of cells there. Those chunks of cells often can act as a chemical signal to the other cells, being like, it's too late for me, but make more of you. We're fair with that? So you could have, you can have some sort of, sorry, I don't need to skip it. You have some sort of internal signal that's like, don't divide until all the chromosomes are attached. But then you also have these external factors saying, we should divide or we should not divide. There's this. Most cells have what's called, well, it's called several things. It's called density-dependent inhibition and anchorage dependency. So anchorage dependence means cells need to be holding on to something to divide. Most cells behave this way. So most cells you're expecting to be part of something. So they need to be clinging on, at least on one side, before they'll divide. If the cell's just floating in goo, it shouldn't divide, okay? So for this little cell body to divide, it needs to be holding on to this little blue goo here, or onto the skin, or onto the liver. It needs to be holding on to something. Most cells also show something called density dependent, actually they're sitting on the bottom of the dish. Density dependent inhibition, meaning if they get too crowded, they stop dividing. Any reason this makes sense? From a practical perspective? Yeah, if you're already full, stop making more, right? And it's not like the cells are broken and they can't make more cells. They just don't want to right now, right? So if you take some of these cells off, right, suddenly there's space, yeah, they'll go and they'll fill back in. We're okay with that? But if they're crowded, they typically stop. If they're crowded, they typically stop. If they're loose and they don't have something to hold on to, they typically stop too. The exception, as the exception is always, the exception is cancer. So you see how these cells right here are behaving nicely? One nice sheet, once they get full, they're done. And these ones are not behaving nicely. They just keep growing. They stack up. They're happy to like squash each other. Cancer cells are super weird, and they can just keep going. There are lots of reasons why they can keep going. Stop, no. What? Sorry. I have a slide of reasons cancer cells can keep going. Here we go. First of all, they don't listen typically when the body tells them no. They're like, la la la, I can't hear you. Not only do they not respond to the break signal, the stop signal, they often will make their own growth factor. They'll be like, I should just encourage myself to grow, right? Cells don't normally do that. Cells normally listen to some other signal to be like, oh, the rest of the organism wants me to grow, I should grow. But no, cancer cells are happy to make their own growth factor. They also can behave as if there's a growth factor, even if there isn't one, which is weird. And they often have just the controls broken, basically, where they just can keep going. So instead of receiving a stop signal if they're not anchored, right? Because cells break off of different parts of the body all the time. Cells break off and they float around. Who cares? Because normally the cell floats around. It can't do much because it's not going to go through mitosis if it's not anchored. But cancer cells can. They can just, go ahead and keep dividing. This is a problem. Because then cancer from one spot can float through the body and find a new home. That's a problem. They can also, this density thing, they don't worry about getting crowded, right? So, sure, I'm making a bunch of me, sure, I'll just make more, right? You'll form up a tumor. And so a lot of cancer treatments are trying to target this, right? Cancer treatments try to stop this happening in different places. A popular one, and I forget the name, I think it starts with a V. A popular one tries to mess around with microtubule formation. Where's a good picture here? Good grief. Oh my goodness. Tries to rest it in something like prophase. Be like, okay, just stop doing that. Something like, oh gosh. Don't even get through this phase. Just you're done. But cancer drugs often target many different parts of the cell cycle. Okay, if you only have brain space for one slide, hopefully you memorize everything, right? That's great. If you only have brain space for one slide, come on. Good grief. Here, this one. You're looking at this. So the order that things happen in, G1, S phase is where synthesis is happening. G2, the M phase, you're expecting a checkpoint at G1 that's going to tell the cell whether or not we're going to divide, period. And a checkpoint at M saying, are we ready to actually divide? We're cool? Great. I will see you all on Monday, bright and early. And yeah.